Okay, in the previous video, we talked about transcription start sites, and we just barely touched on transcription factors. So here I'm going to do a short kind of explanation of transcription factors, and we're going to talk about how they affect transcription and the functionality of the gene within the cell. All right, so first, here's a little picture I pulled off of a paper, which actually I put this paper up in or link it somewhere so that you guys can read it if you'd like. It's not beyond you. I think you can get this one. So let's first talk about the sequence. Let's go to the bottom first, because I think that's the most similar sequence to the one I drew for you before. And so here we have our transcription start site. That would be 0. They didn't put a 0 there, but that's because actually the first one over here, the first nucleotide over here would be +1, and this would be -1. So this is the theoretical in between the two, which would be 0 if it existed. But 0 does not exist as a nucleotide. So there is, I see why they don't put 0 there. But the idea is this would be -20 and this would be +20 letters along the sequence. And about -30 to -35, here you have your TATA box. So this would be where RNA polymerase binds, and it would start copying the DNA and making an RNA right in here. All right, and that's all good and fine. INR initiation, I forget exactly what INR stands for, initiation something, but INR, it's a region that can be identified. There are sequences and proteins can bind here, but this one happens to be right on the transcription start site. Now, there are other transcription factor binding sites. Now, we talked about those. These are places where proteins are going to bind that are going to interact with the RNA polymerase. So the RNA polymerase binds here, and it won't start copying until you get down here. And it's generally a certain distance away, but that can be altered depending on what transcription factors are here and what the shape of the actual DNA, what kind of loops are made in between here, things like that. The DNA can loop back on itself and do all kinds of interesting things here, and that is exactly what happens. One of these that maybe a protein binds here, all right, and another protein binds here. All right, something that, say, binds at the DPE and another one binds at the BREU. And when those bind, those proteins are chemically attracted to one another, and they can bend the DNA over, all right? And as you can see here, that would bend this, and it would bring, as this is brought over, it's going to bring the RNA polymerase to a new place. And as it bends over, as this one is chemically attracted to this one and this one binds here, it brings the polymerase that originally bound here maybe further up here than you saw here. And that would alter maybe where the transcription start site is. So that's kind of how that works. And these transcription factors can bend the DNA and do different things. These proteins can alter the speed, the rate in which RNA polymerase, so something might bind right next to it and allow it to go much faster, or something might bind right next to it and slow it down. Either of these cases are possible: speed up, slow down, and including turn it off completely, as in shut it down so that nothing is made. It can also alter the transcription start site and move it forward or back if there are multiple places that transcription might occur. The bending of the DNA can alter it. There can be more than one TATA box. Actually, TATA boxes, there's generally only one. If there is one, there's only one, but there are other promoter sequences where RNA polymerase can bind, and those are usually the genes that have multiple transcription start sites. So if there's a TATA box there, generally you're under the impression that 90 maybe 95% of the time, you're only going to have one transcription start site. But if you have some of these others, like there's something called a GC box instead of a TATA box. GC boxes, on average, have 3, 4, 5 transcription start sites. So those there's a little bit more going on with a GC box. And actually, I keep saying the most common promoter is TATA, and that's not the case in eukaryotes. The GC box is the most common in eukaryotes. The TATA is the most common in bacteria. So it's generally thought of that the TATA is the most common, but that's really only true of prokaryotes. We have TATAs, but we primarily, as humans especially, have lots of GC boxes. And there are some reasons maybe for that. Perhaps the inclusion of ALU elements specifically in humans have brought more GC boxes to our sequences. Those are transposons, if you didn't know. All right, nobody's proven that. But the idea here is that these promoter sequences interact with the RNA polymerase and begin transcription somewhere nearby. Now, exactly where, you're not always sure, but you need to at least get a general idea, all right, of where it's going to begin. And here's a good thing to know, because you can reduce the amount of lab work you do. And lab work takes time, costs money, and the reason a lot of times you do this bioinformatics work is to narrow down the possibilities. You know it starts somewhere. Well, let's look at the sequence and see if we can narrow those possibilities down so that we don't have to go through letter by letter and try to figure out which one is the transcription start site. Maybe we can figure out which ones to do our experiment on first to see if it does indeed work and is the transcription start site. So that's the real reason for doing a lot of bioinformatics. Now, these proteins here that bind, these transcription factors, that proteins that bind to these transcription factor binding sites, these BRED or the MTE, DPE, INR, all of these, they interact with something called histones as well, histone proteins that form the nucleosome. And the function of these is, this is how your cells essentially turn off genes permanently. So realize that you have many genes within your genome, but every cell doesn't use every gene. The majority of them are turned off. So in that particular cell, a different cell will use a different set of genes. Now, there are some genes that have to be turned on in all cells for them to survive, but there are a large group of them that are unique to each cell type. And depending on which genes you turn on or turn off is what determines the nature of that cell type. Like, are you a liver cell? Are you a neuron? Are you a adipose cell? You know, what type of cell are you? Well, that's determined by what proteins you make, because the proteins determine the chemistry and the physical structure and function of that cell. So depending on which genes you have turned on determines what type of cell you are. And just to give you an idea, a cell that has the wrong combination of genes turned on is really cancer by the most basic definition. So if you start making either the wrong proteins, or you make the wrong amount of them, the wrong combination of them, that's going to confuse your cell. And essentially a confused cell that's not making the right stuff isn't going to be doing the right thing, and therefore technically you can say that that's cancer. It might not be a malignant cancer. It might just be a little odd. But what can confuse cells? Well, mutations can. And you get mutations in the wrong spots. Suddenly that cell starts making the wrong amount of an RNA. Maybe it turns on a gene that shouldn't be turned on in that cell type. And if that's the case, well, then you might develop cancer. It might not be aware. It might just be a little bit of an odd cell. Might not do anything wrong at all in the beginning, but might be the beginnings of something more serious. Now, what is it that turns genes on and off? Well, it has to be how they're wrapped around these histone proteins and forming the nucleosomes. Essentially, without going into detail, because it's a really complex process, think of it as this. If you wrap the piece of DNA where your gene is at, like this right here, and you wrap it around one of these histones very tightly, you essentially are going to turn it off, because when it's wrapped tightly, all these proteins that need to stick all along here can't get there. So a very tightly wrapped piece of DNA around these histones is essentially going to turn it off. If it's in between the histones, all right, these nucleosomes, when you put DNA around the histones, you form a nucleosome. So if you're in a piece of DNA that is in between them, then you're on. Essentially, all these little proteins that stick here can now attach. If, however, you're wrapped around it, you have less chance of sticking, all right, to the DNA. And then if you're wrapped very tightly around it, essentially you're turned off. So, you know, if you're a skin cell, you want all the genes that turn you into a neuron, you want those pretty much turned off. All right, there's some of them are going to be in common, but some of them are not. And the ones that make you a skin cell need to be essentially either unwrapped completely or wrapped loosely. All right, and the ones that make you a neuron, they need to be wrapped tightly so that those mRNAs are not made, and thus the proteins are not made. So this is kind of the way we turn genes on and off, and generally this is done, you know, when you're developing in the womb. A lot of these cells make this decision, and then when they divide, this decision is passed on to the daughter cells. And so, but the decision as to what gets wrapped around these histones tightly and what isn't is typically made at the early developmental stages. All right, now moving up here, what are some of the things that can alter or determine how tightly you get wrapped? Well, one of them is something called a CPG island, and I at least have to mention it. And a lot of genes, especially in humans and in mammals, we have lots of these CPG islands. And you can just think of it this way: these C's in a CPG. Now, a CPG just really is a C followed by a G going along the sequence in the normal way. In other words, the P really stands for a covalent bond. And if you're and not a hydrogen bond going the other way. So a CPG island is a place you get a lot of C's followed by G's, and it's very specific. It's a C followed by a G. A G followed by C is not the same. Actually, it would be in the other direction on the strand, but on this particular piece, going in this direction from left to right, that is a CPG island if you get a bunch of these C's followed by G's. And a C followed by G, at least in mammals and humans, vertebrates actually, these can be what are called methylated. And I don't have time to talk about exactly what that means. That's more advanced class. But if you methylate the C's, you can think of it as making them either sticky or not sticky to the histones. And so you alter the methylation pattern of the C's, and you're going to alter how well they interact, or I shouldn't say more or less sticky to them. I should say just how they interact, because it is different with different sequences and with different histones. So what I will say instead is that when you methylate the C, or don't methylate it, you alter its interaction with how it wraps around the histones, and therefore by altering that interaction, you also alter the access to these regions of the DNA. So of course, if I make all of this region in here, maybe there are a lot of C's and G's here, and they get methylated. Well, if they do, maybe that makes it stickier to the histone, and therefore RNA polymerase and these other proteins cannot get access. And when they can't get access, it means the RNA isn't going to get made, and therefore that protein isn't going to get made. You have essentially turned off that gene. Now for flies, here is the thing and why we're not actually looking at this in this class. Flies don't use CPG islands. They use a different set of sequences. It's a much longer sequence. It's 4, 5, 6 letters. It's not just a simple C followed by a G. And it's a much more complex process in flies. So here's a case where we do it the simple way. We're supposed to be, more advanced or whatever, but, it's all relative. Flies alter their histone binding using a different set of sequences than we do. We use primarily C's followed by G's. There are other sequences that we use. Most people don't know they exist, but we do. And some of them are in common with flies. But as a general rule, we use, as humans anyhow, invertebrates primarily, and higher apes, we use C's and G's. And again, this may be the result of transposable elements. They may have altered this structure here, and actually may be responsible for the reason that we use C's and G's and as our method to turn on and off our genes. This goes way back into evolutionary biology, but what you should understand is primarily you do kind of need to understand this, but you absolutely have to understand where you find the transcription factors. And so here I'm going to tell you right now, transcription factors are mostly, almost, I'd say 90% of them or more, are found before the transcription start site, and probably 80% of them are found actually before the promoter, the TATA box here in this case. Most of them are found over here. But there are some that are found in this region between the where the RNA polymerase binds, the TATA box, and the start. There are more here. And then there are a few that are found after transcription start. All right, now the ones over here are rarer, is that the right way to say that? Or more rare, less common, but they exist. And in humans, I'm not certain about flies, but I know in humans that transcription factor binding sites, these little things here, can be found certainly into the first exon and sometimes into the first intron. But as a rule, you look for them up here first, but it doesn't mean that they can't be after the transcription start site and even into the first, even into the coding region. That means after the ATG, and then further down even into the first intron. Not even the first exon, but into the first intron. Past the first exon and even into the first intron. But as a general rule, after that, you don't see transcription factor binding sites. There are some things that happen around the termination sequence that often have more to do with translation than they have to do with transcription. So generally, we think of these regions are places we look for things that regulate transcription. All right, and so you now know what transcription factors are.